Yogurt is a dairy product made from milk that has been fermented by specific bacterial cultures, including Streptococcus thermophilus. The production of yogurt typically involves several steps, starting from sourcing milk from dairy farms or suppliers. The milk is then pasteurized to kill any harmful bacteria and homogenized to ensure a consistent texture. Next, the milk is inoculated with the bacterial cultures which initiate the fermentation process. After fermentation, the yogurt is cooled and often flavored or sweetened before packaging and distributing it to customers. The main ingredients of yogurt are milk and bacterial cultures. The milk usually comes from cows, although yogurt can also be made from the milk of goats, sheep, or even plant based sources like soy or coconut milk. The bacterial cultures, including Streptococcus thermophilus and Lactobacillus bulgaricus, are carefully selected and added to the milk to facilitate their fermentation. Streptococcus thermophilus is one of the key bacterial cultures used in yogurt production. It is a thermophilic bacterium, meaning it thrives in high temperatures, and this is essential for the fermentation process in yogurt making. Streptococcus thermophilus's shape and arrangement can be determined from its name. Strepto, which gives the arrangement, and coccus, which tells us that it is a spherical shaped bacterium. This particular bacterium is also classified as a lactic acid bacterium. Therefore, the metabolic process used to produce yogurt involves the fermentation of lactose, which is the primary sugar in milk, by the bacterial cultures. This fermentation produces lactic acid, which acidifies the milk and gives yogurt its characteristic tangy flavor and thick texture. It is facultatively anaerobic, meaning it can metabolize both with and without oxygen. And during large scale production, Streptococcus thermophilus is collected from pure culture stocks and inoculated into the milk at specific concentrations to ensure the right fermentation. In small scale or home production, the bacterial culture can be obtained from yogurt starters bought at a grocery store or by using small amounts of previously made yogurt as a starter. Once the yogurt is prepared and ready to eat, the Streptococcus thermophilus bacteria are typically no longer alive. This is because they are sensitive to the acidic environment created during fermentation. Their metabolic byproducts, however, contribute to the flavor and texture of the yogurt. The substrates for lactic acid fermentation are lactose and the bacterial cultures, including Streptococcus thermophilus, and the main product of this metabolic process is lactic acid. There are no specific genetic modifications or genetic techniques used in traditional yogurt production. However, in some cases, genetic engineering techniques may be used to modify the bacterial strains for specific purposes, such as enhancing the yogurt's texture or flavor.
Hello, this is my presentation on my microbes in me paper. I have my notes over here, so I'm just going to be looking at those. For this project, I chose lactobacillus because I thought it would be interesting, and it's something that I see a lot at my work. I work at Planned Parenthood, and we do a lot of vaginal swabs, and we see lactobacillus a lot. First, what is it? Lactobacillus is a gram-positive, non-spore-forming, rod-shaped bacteria. It's aerotolerant and sometimes strictly anaerobic, and all of them are strictly fermentative in their metabolism. And that strictly fermentative quality is really what I feel sets it apart from a lot of bacteria and why it's so useful in so many ways. Some of the ways it's useful are in food production. It's used for fermented foods like cheese, pickled vegetables, yogurt, and then some fermented meats like salami and stuff. And then in us, it plays a big role in both our gut biome and our vaginal microbiomes. And that was what I specifically wanted to look into. And in the vaginal microbiome, it creates substances, the fermentative quality creates substances that are antimicrobial, which help ward off infection. Um, and then one of the fun facts I learned about this when in my research was in a study with over 300 participants uh, in the microbiome, they found a statistically significant difference between bacteria in people's vaginas from different ethnic groups, which I thought was really interesting. Yeah, that's my presentation.
Human microbiota are a cluster of trillions of bacteria that can survive on and within our bodies at their habitats. These microorganisms live very much everywhere on and in our bodies. So, of all human microbiota, let's focus on Staphylococcus aureus. Staphylococcus aureus is a type of bacteria predominantly found on skin and mucous membrane. This bacteria is a non-motile, gram-positive, non-encapsulated, and has a coccus shell. This particular species is capable of undergoing a facultative anaerobic metabolism. The fact that it can utilize oxygen usage indicates that it doesn't necessarily require oxygen in order to survive, means that they can survive without utilizing oxygen. This bacteria is a gram-positive bacteria because it has a purple appearance when viewed via microscope. After being stained with a gram, Staphylococcus aureus is a harmless colony on human skin, but it has a major pathogens that can cause variety of infection, including serious illness such as pneumonia and endocarditis. Metabolically, Staphylococcus aureus is a heterotrophic bacteria, which means it receives energy by break down organic substance in its surrounding. It may also ferment the various kinds of carbohydrate, including glucose, mannitol, and lactose, to produce energy and a metabolic byproduct. Staphylococcus aureus is a type of bacteria that plays a significant role in maintaining the ecology of human body and skin. It is usually present as a commercial bacterium that helps in supporting skin healthy and protect against harmful bacteria.
Presenting my homework assignment on microbes in me, specifically the microbe Lactobacillus crispatus. The human microbiome consists of all of the microorganisms, like bacteria, archaea, fungi, and more, that live in or on humans. Microbes accumulate in our bodies from birth, and the species of microorganisms we host vary between organs. Microbes play an essential role in the development and physiological activities of humans. Many of these microbes are essential for human health. Microbes found on humans are capable of producing essential vitamins, extracting nutrients from foods, aiding our immune system in recognizing foreign microbes, and producing anti-inflammatory compounds to fight off other harmful microbes. Many factors can affect the specific microbiome of different regions of the human body. Humans are exposed to complex microbial communities within their external environments. Maternal microbiota affects the microbiota of infants. Antibiotics can change microbial compositions, and diet greatly affects the human microbiome, specifically the gut microbiome. Disease states can result from compositional changes in microbiome speciation or abundance changes within microbiome species, both resulting in microbiome dysbiosis, or expression of virulence characteristics by a species within the microbiome. In 1876, Robert Koch established that a specific bacterium could cause a specific disease, supporting the germ theory of disease. Joseph Lister introduced the use of chemical compounds, or antimicrobial agents, to significantly reduce the presence of microbes in 1867. Antimicrobial agents like antiseptics, disinfectants, antibiotics, antivirals, antiparasitics, and antifungal drugs can be used to destroy or inhibit microorganisms in order to control which microorganisms we interact with to prevent disease. The vaginal microbiome of the human vagina hosts a complex mixture of microbiota that changes with the menstrual cycle. The vagina produces an environment that is wet, dark, acidic, with a low pH, and inconsistent for microbes. The vaginal microbiome is largely dominated by a single genus, Lactobacilli, that cluster into 5 core groups dominated by Lactobacillus inners, Lactobacillus crispatus, Lactobacillus gasseri, or Lactobacillus gseni, and a fifth group that had higher proportions of strictly anaerobic organisms. The acid-tolerant lactobacilli ferment glycogen that is produced by the vaginal epithelium, which forms lactic acid and causes a low pH in the vagina. This production of lactic acid, combined with hydrogen peroxide and bacteriocins, protects the vaginal environment from invasion by other microorganisms and infection of the vaginal epithelium, and has been associated with an overall well-being. The vaginal microbiome is dynamic, and its composition changes in response to pregnancy, menstruation, external influences like douching and sexual activity, and disease states. An imbalance of the vaginal microbiota, typically a loss of lactobacilli or any other dysbiosis in the vaginal microbiome, correlates with bacterial vaginosis. Imbalances of the microorganisms of the reproductive tract can cause disease, infertility, miscarriage, premature delivery, and gynecological tumors. Lactobacilli are an abundant and diverse group of lactic acid bacteria that reside in the oral cavity, gastrointestinal tract, and genitourinary tract of the human microbiome. Lactobacillus crispatus is a species of lactobacilli that is found in human feces and the human vagina. It can make up more than 80% of all vaginal bacteria and is one of the most active species in a healthy vagina. Lactobacillus crispatus is an obligately homofermentative, catalase-negative, gram-positive bacterium. When the dominant bacteria are Lactobacillus crispatus and or Lactobacillus gseni, most of the metabolites in the vagina are amino acids and dipeptides, such as higher levels of ornithine, lysine, glycoproline, and phenylalanine. Lactobacillus crispatus maintains vaginal health by producing acids, hydrogen peroxide, and bacteriocins in the vagina, having an inhibitory effect on common pathogens like vaginitis, having good antioxidant properties, preventing the colonization of HeLa cells by opportunistic pathogens, protecting against STIs, accelerating the re-epithelialization of vaginal epithelial cells, and increasing secretion of vascular endothelial growth factor A. The absence of Lactobacillus crispatus is associated with bacterial vaginosis, and certain strains of Lactobacillus crispatus can be used as biotherapeutic agents in order to reduce recurrent urinary tract infections and BV. Thank you.
Within our bodies, we hold giant galaxies made up of trillions of microscopic organisms that grow, coexist, and outcompete each other. These microorganisms live in close symbiosis with us and play a fundamental role in controlling most aspects of our physiology. They break down foods we are unable to digest, producing valuable energy sources. They also produce vitamins like vitamin K and B, as well as amino acids. Perhaps one of the most powerful symbiotic relationships the microbiota has is the one it has with our immune system. It eliminates viral infections and regulates inflammation. This symbiosis can be disrupted, and contrasting with the word symbiosis, we have the word dysbiosis, which refers to a change in the quantity and quality of the microbial communities within our body that is associated with disease. Here are some factors influencing that imbalance. If you're a microorganism, getting to live in the human gut is much like stumbling upon Earth while traveling through the Milky Way galaxy. It is a nutrient-rich environment brimming with life. We're going to focus in on a genus that lives in the colon, the largest part of the intestine that's pictured here. And if you're a microorganism that uses complex carbs, this is the place to be. It is the reason Bifidobacterium loves it here. The gene content of this genus is reflective of its metabolic adaptation to the complex carb-rich environment of the colon, as it encodes a large number of carb-modifying enzymes that break down non-digestible carbs into short-chain fatty acids, vitamins, and bioactive peptides, all needed to keep the host running optimally. Species members of this genus enrich the environment of the colon in a number of ways. They inhibit the growth of cancer cells by altering physiological conditions in the colon. They fight off infections by colonizing the colon and outcompeting those pathogens, and they regulate pro-inflammatory signaling pathways that reduce inflammation. And I'll leave you here with this slide that shows some additional benefits that different species members confer upon the colon.
In this video presentation, I will be sharing my understanding of a microorganism and how it interacts and affects our daily lives. Can you guess it? A germ walked into a bar. The bartender said, "We don't serve your kind here. You'll have to leave." And the germ said, "But I work here. I'm staff." If your guess was Staphylococcus aureus, your guess was correct. Let's begin with the history. In 1871, von Recklinghausen was the first to observe and identify Staphylococcus. In 1880, Alexander Oxen discovered and confirmed Staphylococcus. In 1884, Rosenbach separated two strains known as Staphylococcus aureus and Staphylococcus epidermis to study the differences in characteristics. Staphylococcus aureus is a round golden cluster of bacteria ranging from 0.5 to 1.0 micrometers in diameter. It stains purple, indicating that they are gram-positive, and stains golden when they are grown on blood, agar, or salt agar plates. On average, Staphylococcus aureus is found in harmless clusters in the nasal passages and is one of the most common occupants of humans. Many individuals are asymptomatic as they are living and reproducing on every square inch of your body. While some strains of Staphylococcus aureus can become resistant to multiple antibiotics, the strains vary widely in their capacity to make toxins and harmful enzymes. Characteristics are pathogenic spherical cells, non-motile, facultatively anaerobic, non-spore forming, catalase positive, oxidase negative, and coagulase positive. If you didn't know, now you know. Laughter is the best medicine. Doctor, we got the results back. There's staph in your nose. Me, I don't remember hiring anyone. Pathogenicity results from three features: structures that enable it to invade phagocytosis, the production of enzymes, and the production of toxins. Transmission is through aerosol. Some diseases, depending on the site of infection, can cause pneumonia, wound infections, food poisoning, toxic shock syndrome, in addition to diseases of the joints, bones, heart, and blood, and minor skin infections such as impetigo and boils. Thanks for watching.
Okay, so miso. It's delicious. It's a fermented bean paste, super traditional in Japan. You've probably encountered it in miso soup, but it's full of umami flavor and salt, and it's really cozy on a rainy day. Miso is made with a two-step fermentation process that starts with inoculating a grain substrate, usually rice, more rice than this, whatever, with spores of this mold Aspergillus oryzae, Japan's national mold, which is a filamentous fungus. Grown on a plate, it kind of looks like regular kitchen mold, but unlike regular kitchen mold, it does not produce aflatoxins and is safe for food production. So spores, more spores. The inoculated rice is left in a warm, humid place to incubate for a couple days, turning it every 12 hours to make sure that A. oryzae has enough oxygen. A. oryzae is an obligate aerobe, and to encourage growth and maintain an appropriate temperature. After a couple days, you have this extremely moldy rice called koji, and A. oryzae has secreted a bunch of hydrolytic enzymes into the koji. It can break down the rice starch into sugars that can be fermented, and it can break down proteins into amino acids. At this point, you move on to the second stage, where you take the koji and you mix it with mashed soybeans, salt, and a halotolerant bacteria and yeast, pack it tightly into a fermentation vessel, and let it ferment for a period of time from like several weeks to maybe a couple years. At this point, A. oryzae doesn't grow anymore. It's in this anaerobic, salty environment, but the yeast and the bacteria are very happy fermenting away, producing sugars and alcohols and organic acids. The enzymes from the koji are still happy breaking down starch and proteins and generating amino acids. Organic acids and alcohols are combining to form esters. There's just a lot of stuff going on to make complex flavors and aromas. And you let it continue like this until it tastes the way you want it to, it smells the way you want it to. At that point, you heat it up to stop the microbial activity, store it, cook it, make some soup, make some salmon, bake some cookies, whatever, eat it, yum. And these are my image credits.
Here is an overview of Lactobacillus acidophilus in the gastrointestinal system. When looking at the phylogeny of Lactobacillus acidophilus, it is a bacteria and its phylum is a Firmicute, which is usually found in the gastrointestinal system. Its class is Bacilli, the order is Lactobacillus, family is Lactobacilliaceae, genus is Lactobacillus, the species is Lactobacillus, and the subspecies is Acidophilus. Some characteristics of this microorganism is that it is gram-positive, it is rod-shaped, and it's a facultative anaerobe. It can grow in aerobic environments, but it does prefer anaerobic environments. It's resistant to acid and bile, so it can live in the gastrointestinal system. It is also tolerant to low pH. And some of the characteristics of the ecosystem that it lives in is that the gastrointestinal system is slightly acidic. It's around body temperature because it's in the body, and it is moist. Some functions of Lactobacillus acidophilus is that it contributes to the host and microbial communities, and this is usually done by inhibiting the growth of pathogens, and this is done by reducing the pH, which is through fermenting and producing D-lactic acid. This microorganism also improves lactose metabolism in the body.
Kimchi is a staple dish in Korea. Isn't just delicious? It's a world teeming with tiny microorganisms known as microbes. These microorganisms work their magic, transforming ordinary cabbage into flavorful kimchi to enjoy. In this exploration, we will delve into the scientific aspect of kimchi, examining the specific microbes involved and their use of unique process called lactic acid fermentation to enhance kimchi flavor. This essay will uncover the fascinating microorganism behind this type of food, shedding light on the organism involved in this fermentation process. The creation of kimchi relies on a well-coordinated partnership among various microorganisms, primarily known as lactic acid bacteria. This bacteria, renowned for their ability to convert sugar into lactic acid in environments without oxygen, are crucial for the fermentation process. essential in kimchi production. Leading this microbial ensemble is Lactobacillus kimchi. It's a rod-shaped bacterium uniquely adapted to the environment of kimchi.
Approximately 1/3 of solid waste consists of microscopic remains from trillions of microbes residing within and on us. Human microbiota comprises bacteria, fungi, and other organisms vital for digestion, immunity, and possibly mood regulation. Research suggests microbiota influence brain development and function through the gut-brain axis. Factors like diet, exercise, medication, and stress affect gut microbiome composition. Dysbiosis, an imbalance in gut microbiota, is linked to various health conditions. Emerging therapies like fecal microbiota transplantation aim to restore microbial balance. Alterations in gut microbial composition are linked to mental health disorders. The human gut hosts a diverse microbial community crucial for well-being. Beneficial bacteria like Bifidobacterium longum aid in digestion and produce short-chain fatty acids SCFAs. B.longum belongs to the Bifidobacterium genus, which is a member of the Actinobacteria phylum. It is anaerobic, meaning it thrives in environments that lack oxygen. One of the unique features of B. longum is its adhesive-like properties, which allow it to adhere to the intestinal wall lining. This allows easier colonization within the gut. SCFAs nourish the gut lining, regulate metabolism, and inflammation. Disruptions from factors like processed foods or stress can lead to digestive issues and diseases. Factors like dietary fiber intake, physical activity, and sleep patterns influence gut microbiota. Gut microbiota may modulate immune responses and influence susceptibility to diseases. Manipulating the microbiome through dietary and lifestyle changes holds potential for preventive and therapeutic medicine. Exciting advancements are anticipated in harnessing microbiota for health optimization. Microbiota modulation affects mental health outcomes via the gut-brain axis. Differences in gut microbiota between societies contribute to health disparities. The microbiota shapes individual, societal, and environmental health.
The human microbiota refers to the collection of microorganisms associated with a particular environment of the body. These microbiota are part of the larger human microbiome, which includes everything from the individual herself to the environmental factors surrounding. The microbiome as a whole is individualized to the host and includes microbes that can both harm and help. To get a better idea of the ways in which microbiota affect health, we'll focus in on the vaginal ecosystem and the role Lactobacillus acidophilus plays within it. The vagina itself is a warm, varyingly acidic, relatively anaerobic, and moist environment that plays host to a limited range of microbial communities. A healthy vaginal microbiome is hard to define, as it's dependent on a wide variety of many factors, but most healthy ecosystems are comprised heavily of lactobacilli, which, through the production of lactic acid, act to keep the pH of the vagina low and inhibit many other invading microbes. L. acidophilus, in particular, is considered a low guanine cytosine gram-positive bacteria and belongs to the phylum Firmicutes. These microbes rarely cause disease. Instead, they protect the body by inhibiting growth of pathogens, a situation called microbial antagonism. Most L. acidophilus are microaerobic, but grow best in anaerobic environments. They prefer temperatures between 35 and 38 degrees Celsius, pH levels around 5.5 to 6, and they are incredibly resistant to acids and biles. They can ferment glucose, fructose, and lactose in order to produce large quantities of lactic acid, making it one of the major players in the maintenance of healthy pH levels in the vaginal ecosystem. Several factors can suppress L. acidophilus function, from hormone changes to diet, which can in turn affect optimum pH levels. This gives other opportunistic pathogens like G. vaginalis or Candida the opportunity to grow at accelerated rates, causing a range of issues, from yeast infections or bacterial vaginitis to more serious issues like pelvic inflammatory disease or infertility if left untreated. In this way, we can start to understand the complexity of the vaginal ecosystem and its microbiota, and how small changes can result in significant health impacts.
Hello, and welcome to Microbiomes and Me. We live in symbiosis not only with each other and the environment, but within our bodies. The human microbiome is a diverse community of microorganisms that inhabit various parts of the human body, including the skin, mouth, gut, and various mucosal surfaces. The human biome interacts closely with our immune system and plays essential roles in digestion, metabolism, and protection against pathogens. Disruptions in the balance of the human biome, known as dysbiosis, have been linked to various health conditions. The microbiome of the sinuses typically includes bacteria, fungi, and occasionally other microorganisms. They help regulate the local immune response and prevent the overgrowth of harmful microorganisms. Throughout inhalation, the meatises of our air passages come into contact with pollutants, airborne allergens, and microorganisms. Due to the nature of the sinuses being warm, dark, and humid, this becomes a fertile breeding ground for pathogens. Staphylococcus aureus is a gram-positive facultative aerobe and plays a major role as a commensal bacterium and can become a harmful human pathogen. Sinusitis, also known as sinus infection or rhinosinusitis, is a very common condition where the paranasal sinuses are inflamed, causing a congested nose, headache, and facial pain. The microorganism most frequently associated with these cases is S. aureus. Sinusitis can be caused by infection, anatomical abnormalities, and seasonal allergies. This affects 1 in 8 people living in the United States. The mucus drains into the nasal cavity through small openings, and when the sinus is blocked, fluid builds up, making it a favorable environment for bacteria or viruses to grow and cause infection. Typical sinusitis symptoms are described as dull pain or constant pressure, commonly localized to the affected sinus. Allergic rhinitis is caused by allergens such as pollen and pet dander. S. aureus releases superantigens capable of triggering T cell activation, leading to an exaggerated inflammatory response that may become unmanageable and resistant to treatment. The most widely agreed upon solution is prevention through proper body hygiene as well as home hygiene. People with deviated septums and nasal polyps seem to be more susceptible to allergic rhinitis and may require surgery. Overall, S. aureus is a versatile bacterium that can exist as a harmless commensal or a dangerous pathogen depending on the circumstances. Its ability to cause a wide range of infections and its propensity for antibiotic resistance make it a significant public health concern. Thank you.
The human microbiota. The human microbiota consists of the billions of microbial cells living in and on each person. It was first conceptualized in the 1600s with the work of Antony van Leeuwenhoek and his early microscopes. Some consider the human body to be a superorganism, meaning a combination of human and microbial cells working together. We rely on these microbes to protect us against pathogens, regulate the immune system, and break down nutrients, and ultimately help us maintain homeostasis. Next, we'll take a closer look at the skin microbiota in particular. Skin is colonized during birth. It includes a diverse array of microbes that inhabit distinct ecological niches. Skin is acidic, cool, dry, nutrient poor, and it undergoes constant desquamation, making it difficult for microbes to adhere. As a result, skin microbes are highly adapted to thrive under these conditions. We rely on the skin microbiota for healthy skin development and immune function. It also provides colonization resistance, meaning it prevents the growth of pathogens. Dysbiosis of the skin leads to various diseases ranging from acne to serious wound infection. One of the most prevalent microbes found on the skin is the bacteria Staphylococcus epidermidis, which are small gram-positive facultative anaerobic cocci arranged in clusters. It's halotolerant, it can grow in low relative humidity, and it's an expert biofilm former. It's closely related to Staphylococcus aureus, the cause of MRSA infections, but it's far less virulent. It promotes skin homeostasis by educating and priming the body's immune system and contributes to colonization resistance. However, qualities that make Staphylococcus epidermidis a successful colonizer of the skin also contribute to its success as an opportunistic pathogen.
The human microbiota in Staphylococcus hominis. Staphylococcus hominis typically appears as spherical cells, cocci, arranged in clusters resembling grapes when viewed under a microscope. It is non-motile and does not form spores. They are gram-positive. They typically colonize areas rich in apocrine glands, such as the axilla or pubic region. Due to the salty nature of sweat, they are halotolerant. Staphylococci are facultative anaerobes, meaning they can grow in oxygen-rich and poor areas. While they are typically a commensal bacteria, they have been known to be opportunistic and cause infections. This often happens in individuals who are immunocompromised. Body odor is often thought to come from sweat. However, the odor comes from the bacterial breakdown of apocrine secretions, or sweat. S. hominis is one of the bacteria contributing to this interaction. S. hominis contains the enzyme called CT lyase. This enzyme produces thioalcohols, which give sweat the pungent odor. S. hominis encodes a proton-coupled oligonucleotide transporter that imports the thioalcohol conjugated precursor into the cell, and subsequent catabolism results in the foul-smelling thioalcohol. S. hominis plays an interesting role in the human microbiota. While the effects are not detrimental to health, they are often unwanted.
Welcome to my exploration of the human microbiota, where trillions of microorganisms call our bodies home. Today we'll journey through the intricate world of our microbiota, focusing on the gastrointestinal tract and the fascinating role of Bacteroides fragilis within this community. Our bodies are teeming with microorganisms, ranging from bacteria and archaea to eukaryotes and viruses, collectively known as the human microbiota. These microorganisms find themselves in various regions of the body, together playing roles in host health, from aiding in digestion to nutrient absorption to modulating the immune system and defending against pathogens. One region that is pivotal in nutrient uptake and immune defense is the gastrointestinal tract, which contains the largest concentration of microorganisms within the entire human body. While it primarily contains microorganisms within Bacteroidetes and Firmicutes, the gastrointestinal tract contains other microorganisms from over 50 different bacterial phyla. All of these shape our health in profound ways. This anaerobic region of the body contains many obligate anaerobes and facultative anaerobes, but the exact composition is influenced by a variety of factors, including partial oxygen pressure gradients resulting from GI colonization in the neonate and fluctuations in pH as a result of dietary intake. Of course, disruptions like dysbiosis from prolonged antibiotic use can lead to serious health issues, such as Clostridium difficile infections, so it is not without its post-health risks. Today, I'd like to focus on Bacteroides fragilis, a prevalent obligate anaerobe gram-negative bacillus living within the human colon, often misunderstood as solely pathogenic. B. fragilis, while considered the most virulent species of Bacteroides due to its ability to escape the colon via the bloodstream and cause infections, is not just a troublemaker. It plays a crucial role in our well-being. For instance, it ferments carbohydrates, yielding essential fatty acids vital for our energy needs. Moreover, B. fragilis produces polysaccharide A, a potent immunomodulator that safeguards against inflammation and disease. In summary, while B. fragilis carries risks, its multifaceted contributions are representative of the symbiotic relationship between a human and its microbiota.
Hello, my name is Heather, and welcome to my Microbes and Me presentation. In this presentation, we are going to go over the human microbiota of the skin flora and the microorganism Staphylococcus epidermidis. So let's get into the human microbiota. The human microbiota is a set of microorganisms inhabiting and interacting with the human body. It is classified into gut, oral, respiratory, and skin microbiota. It consists of bacteria to viruses to fungi and many other microbes. It is essential for human development, immunity, and nutrition. There are many factors such as lifestyle choices, antibiotics, and other chemicals that can impact the human microbiota either beneficially or detrimentally. Within this human microbiota is the skin flora. The skin is home to millions of bacteria, fungi, and viruses. In addition, the skin microbiome consists of several abiotic factors. Some of these factors is that the skin is a dry surface, is aerated, it has a pH of around 5.6, it is rich in lipids and sebum, and it lacks metabolites. There are many roles in the skin's microbiota plays. However, generally, the skin microorganisms have essential roles in protection against invading pathogens, the education of the immune system, and the breakdown of natural products. Along with these roles, the skin serves as a physical barrier, and when this barrier is broken, when the balance between conventional and pathogens is disturbed, skin disease or even systemic disease can result. On this page is a picture of the skin's flora. Now let's take a look at one of the most abundant bacterial colonizers of the skin, Staphylococcus epidermidis. On the left shows the phylogeny of Staphylococcus epidermidis. However, I really want to get into the characteristics and the roles of this microorganism. As a result, this microorganism is catalase positive, coagulase negative, gram-positive, facultative, anaerobic bacterium. In addition, it's heterotrophic and forms biofilms. This microorganism plays many roles within the skin's ecosystem, such as preventing the overgrowth of harmful microorganisms, maintaining a balanced microbial community, forming biofilms, and immune modulation. In addition, these roles within this community, Staphylococcus epidermidis plays its part in microbial diversity and stability, interspecies interactions, and disease susceptibility. As you can see, this microorganism is very important and plays many essential roles in keeping the skin flora healthy and balanced. This is the conclusion of my presentation. Thank you for listening.
Hi, my name is Katie, and I chose the microbes in me topic. The human microbiota is like a world filled with tons of species and environments. Each person has a unique microbiota determined by one's DNA. Here you'll find species of fungi, bacteria, viruses, and parasites. With a healthy microbiota, there's a symbiotic relationship with the host and a diversity of microorganisms all working to maintain homeostasis. However, diet, environment, and medications such as antibiotics can create an imbalance or dysbiosis, causing inflammation. Now let's zoom in on the most densely populated city on our planet, the gastrointestinal tract. Your GI tract hosts anywhere between 10 to 100 trillion microbes, about 2 pounds of microbes. The pH levels vary throughout the GI tract from low to high acidity, and it is very moist and habitable for microbes. The main communities that exist in the GI tract include bacteroidetes, firmicutes, and proteobacteria. Now we're going to hone in on a specific member of the proteobacteria community, Escherichia coli, or E. coli. E. coli typically lives in the GI tract, specifically the intestines. It's rod-shaped, gram-negative, and is able to grow in both aerobic and anaerobic environments, otherwise known as facultative anaerobe. While E. coli is often thought of in relation to illness, not all strains are pathogenic, and many are harmless. Some are even beneficial to the GI tract, as they can aid in digestion, protect humans from harmful microbes, and because they consume oxygen that make the GI tract more habitable for other microbes. E. coli is known for being genetically promiscuous, and it can exchange genes with other strains and even other types of bacteria. Pathogenic strains of E. coli disrupt the function of cells in the intestines, causing diarrhea when they inhibit cells from absorbing water or influence cells to release water in the intestine.